Today, 1st of June 2020 in the USA, Canada, and 2nd June 2020 in Australia, it's my pleasure to welcome Professor Janusz Rak. He is a professor of Pediatrics, Experimental Medicine, and Biochemistry at McGill University in Montreal, Canada. He's also a Jack Cole Chair in Pediatric Hematology Oncology and investigator with the Research Institute of the McGill University Health Center. Janusz previously trained in Poland, United States, and Canada. And for the past several years, his lab has been investigating how oncogenes deregulate stromal and vascular responses in cancer. His current work focuses on EV-mediated intracellular communication, including the regulation of homeostasis, angiogenesis, and brain tumor progression, including in children. Janusz is well known in the field. He published over 150 scientific papers with over 22,000 citations, and he's currently supported by grants from Canadian Institutes of Health Research and other agencies. So please welcome Janusz.
Welcome. Thank you, Carolina, for this kind introduction, and hi, everybody. Thank you for joining us for this discussion. So now I should probably start my presentation. Okay, so basically what I wanted to share with you over the next little while is some of our interests in one particular question, or exploring one particular question, and that is, what are the extracellular consequences of oncogenic transformation? And I titled this, as you can see here, as Extracellular Oncogenes in Pathology of Tumors Trauma, because I think this is something that we haven't fully understood and which may have certain practical and scientific implications. So before I begin, I'd like to, of course, briefly mention members of my group, and of course prior trainees and colleagues who have been in our lab for years sometimes and contributed to this work. And I also wanted to mention that we've had a number of great collaborators without whose help we would never been able to function. Of course, granting agencies paid for all this. So the fundamental question that I think many people ponder, is that, what is the significance of cancer as a disease worldwide? And I would argue that in the midst of COVID-19 pandemic, we should not lose from sight that there's actually an ongoing cancer pandemic, which is signified by something like 9.6 or perhaps even more million people who die every year because of different forms of cancer. Of course, this has been an eternal question for many generations of scientists, and I think the fundamental problem that people have been grappling with is what really converts a normal tissue that has a precise mechanism of action in which cells are fine-tuned and well differentiated, such as the one on the left, what converts it to a violently aggressive tumor that, such as the one shown on the right, glioblastoma, which usually brings up a negative outcome within something like 15 months. So this is a disease that kills people essentially in 100% of time. And of course, what actually is between these two states, seemingly so separate. And I would like to put forward an argument that we have been conditioned to think of tissues and their function in very cell-centric fashion, namely that we attribute the cellular programs, we attribute the function of the cells and their clusters and populations to cellular programs that operate within the cellular milieu. And this was a powerful paradigm and very helpful in many ways, in that it allowed us to focus our thoughts on what actually happens in transition to cancer. And as you can see from this diagram, what's been happening over the last 3 decades is the accumulation of evidence that, in fact, what converts normal to abnormal, or from normal to cancer, are permanent genetic or epigenetic alterations that change the inner workings of cells and convert them into cancer cells. And essentially, these gain-of-function, loss-of-function mutations are responsible for all so-called hallmarks of cancer that we know about. And so, of course, this is a very cell-centric view, because it claims, as it were, or it implies that inner workings of cancer cells actually explain the disease as we know it. And this point of view clashes with the subject we are all interested in, and that is that cells project their molecular content and cargo and information outside of themselves. And one of the most interesting and complicated ways in which this is happening is through production, release, or circulation of extracellular vesicles, which, as we know, contain a great repertoire or vast repertoire of molecular cargo with biological activities that we still don't fully understand. And of course, the complexity of this communication system has been compounded by the fact that there's now multiple classes of vesicles that we know, and I don't want to get into any details of this because I'm sure we've talked about this during these sessions and elsewhere. But it's important to keep in mind that vesicles or extracellular vesicles of all kinds have certain unique attributes, such as they reflect the parental cells, for example, protect their molecular cargo, circulating biofluids, and importantly interact with target cells, thereby mediating the intercellular communication. And of course, in the context of cancer, it may arise our interest, and it's been studied extensively the question, what the processes affects of cellular vesiculation, what processes change the repertoire and biological activity of vesicles. And we know of various influences such as microenvironment, cell death, drug treatment, epigenetics, signaling and cellular regulation. But what I wanted to concentrate on for the remainder of my comments is one particular type of influence, which is not outside in, but is inside in, and this is the oncogenic transformation. So what makes us think, what makes me think that oncogenic transformation has anything to do with the vesicle-mediated intercellular communication? So, of course, I've said this many times on different occasions, and I'd like to repeat it once again, is that very simple cell systems or models can sometimes be very informative in terms of the degree to which a particular process can influence events such as vesiculation. In this case, this is a glioblastoma model where indolent cells can be engineered to express highly oncogenic epidermal growth factor receptor variant 3 oncogene. And then we can ask a very simple question, knowing that certain pathways of vesiculation, either in the form of exosomes or microvesicles, have already been mapped to some extent, and their respective regulators have been identified. What is the influence of this oncogenic transformation on the levels, at least, of these different regulators? And one finds that, indeed, this is not an innocuous transition. So oncogenic transformation changes a great deal in terms of the repertoire of these regulators of vesiculation, either upwards or downwards. And some of the canonical vesicular proteins, such as tetraspinins, for example, CD82, are very profoundly downregulated by expression of this mutant oncogene. And of course, this doesn't end there. There's the whole repertoire of vesicles is profoundly changed to the extent that there's a set of something like 200 completely new proteins that are entering the vesicles as a result of oncogenic transformation. And of course, this is not just a tissue culture artifact generated by this particular system, artificial it might be, because when we look at the real tumors and their transcriptomes, especially in the context of brain tumors such as glioblastoma, we know that these tumors differ in terms of molecular makeup and oncogenic circuitry, and this divides them into several subtypes such as proneural, neural, classical, and mesenchymal. And these subtypes contain different levels of transcripts for different vesiculation regulators, such as shown here. Now, this may be very interesting and is definitely worth pursuing, and we, of course, and many others have taken interest in these changes in regulation of vesicles by different signaling and oncogenic circuitries. But what is really tantalizing for me about this whole process is the fact that we and others have stumbled upon a property of vesicles that changes the current concepts of oncogenesis in a rather interesting way. And namely, we've been conditioned to think that oncogenes operate within the cells, as I mentioned earlier, within the cell confines. But we and others have found that, in fact, cells producing vesicles as a result of oncogenic transformation also carry the very same oncogenes in their vesicles in an active and biologically intact form, which suggests that these oncogenes may actually not only exist within the cells in which they mutate, but also outside of them, and in fact potentially be carried into blood and serve as a biomarker of cancer progression, and in fact be transferred or interact with target cells. So this is a very unique to cancer cells, that these mutant oncogenes can be exported in such a fashion. And proteins oncoproteins are not the only form in which oncogenes can exit their parental cells. Studies have shown that also transcripts of certain oncogenes, such as EGF receptor variant 3, exist in the form of vesicles, as well as genomic DNA, which of course raises questions as to the possibility whether there is any horizontal transformation of target cells produced by this particular process, and whether or not we can use this process as a platform for liquid biopsy. Now, it is instinctive or intuitive that one may think that transformed cells will produce vesicles carrying oncogenes such as EGF receptor variant 3, and this would be a homogeneous population of vesicles, as these cells uniformly express high level of the oncogene. So why would vesicles not contain this particular oncogene? But in fact, nanoflow cytometry and other high-resolution methods have allowed us to actually examine this question in more detail and find that, in fact, this is not the case. That even cancer cells, which are clonal and produce high levels of oncogenic proteins, produce different subsets of vesicles, of which some will contain the active oncogene, while others will contain other proteins and not this particular oncogene, and they may have very different functions than those that produce oncogenic, those that contain oncogenic EGF receptor. I think this is important to keep in mind as we pursue our studies on different activities and resolve these different activities that tumor vesicles may exert on their different target cell populations. But before we understand that in a greater detail, it's probably useful to ask, what is the consequence if EGF receptor containing cell encounter a target cell that doesn't have that oncogene? And many years ago, we've conducted some experiments that sort of aim to elucidate this, at least to some degree. And so what we chose to do is to take EGF receptor expressing cells and incubate them in the presence of vesicles containing EGF receptor oncogene, incubate these vesicles with indolent cells that don't express this particular receptor. And just to make the long story short, what we found is that this particular interaction or entry of these vesicles into indolent cells, which is demonstrated by fluorescent transfer of oncogen associated fluorescence, leads to rather striking biological changes in the recipient cells, namely these indolent cells, which have a fairly low transformed potential as measured by soft agar conformation, can acquire a much higher transformed potential by simple fact that they have been incubated with this particular material. So the oncogene transfer actually changes their biology in a rather interesting and dramatic way. And of course, this also happens in vivo to some extent, although what is the extent of it and what are the exact implications is still under debate. So we've done an experiment many years ago in which we mixed these indolent green cells with red aggressive cells, and the red ones had EGF receptor variant 3 expression. We asked, what would be the outcome of the mixed tumor growth? And one would expect that the mixed tumors would grow somewhere in between these different curves, meaning that the indolent cells would form very sort of slow growing or even dormant tumors, whereas the aggressive cells would grow with their own pace. But in fact, this is not what happened. The actual mixed tumors grew faster than either of the two populations alone, suggesting that there is some kind of contact or communication or interaction between these populations that change their in vivo properties. And in fact, we've observed the transfer of EGF receptor between these populations in tumors in vivo. But these are interactions between tumor cells and tumor cell populations. And I did say at the beginning that we wanted to examine the impact on tumor stroma. So let me just move to some of the thoughts on that particular topic. And what do we mean by tumor stroma? Of course, depends whom you ask. And for me, I think different tumors may have different stromal compositions, and different cells may play different roles. What is almost invariably found in tumor stroma is the vasculature, which is essential for metabolic activity, many other functions of not just normal cells, but tumor cells. And one can see from this image, which is the image of brain vasculature, that the area occupied by tumor is, of course, filled with vasculature, but this vasculature is actually very, very different. So what makes this vasculature different? So what in the other words, do tumor cells produce or do to the vascular environment in which they live? And of course, you know, from the point of view of our discussion today, I would like to focus some of these questions on extracellular vesicles. So in the other words, the vascular cells, endothelial cells, how do they actually respond to vesicles emanating from tumor cells expressing mutant oncogenes such as EGF receptor, for example? So we've done this experiment, and the first interesting thing was that endothelial cells are actually very avid uptakers of vesicles from cancer cells, including vesicles containing EGF receptor, and this EGF receptor oncogene protein can be detected on the surface of endothelial cells exposed to this material. In fact, this is not a completely inert process, because, as might be expected, some downstream targets of EGF receptor become activated in endothelial cells, such as, for example, MAP kinase phosphorylation and AKT phosphorylation. And surprisingly, some of the genes that normally endothelial cells are either don't express or express at very low levels become activated, as if these were cancer cells. So, for example, VEGF becomes expressed, or vascular endothelial growth factor becomes expressed from within endothelial cells that have taken up vesicles carrying EGF receptor. Of course, this has implications for different inhibition and experimental therapies, and even human therapies, whereby we would expect that there would be a greater effect on this particular process by agents acting from within endothelial cells rather than, for example, neutralizing antibodies. And of course, the question is, how does this compare to more sophisticated models of tumor vascular interactions or tumor growth? And one of these models is shown here. So as I mentioned earlier, glioblastoma is now divided into all these different subtypes in which we know a little bit more now, or I would say maybe a great deal, in fact, about their oncogenic mutations, which are listed here, and their transcriptomes are very, very different. And so consequently, one would have thought that tumor cells that initiate growth of these different tumors would also be somewhat different, and they are. So in fact, so far, two populations of so-called glioma stem cells, or glioma initiating cells, have been identified, and by transcriptomic analysis, they either resemble proneural tumors or they resemble mesenchymal tumors. And the proneural glioblastoma stem cells don't express EGF receptor, whereas mesenchymal stem cells do. They express, in fact, high levels of EGF receptor, and often they express a mutant EGF receptor as well. So what do these different glioma stem cells communicate by vesicles to endothelial cells? Endothelial cell circuitry during angiogenesis is well mapped, and there's many players here, molecular players, but how does this interplay respond to the presence of EGF receptor containing or not containing extracellular vesicles from glioma stem cells? And so some of our early attempts to answer these questions are shown here. So initially we compared starved endothelial cells in terms of their growth capacity in the presence of vesicles from either mesenchymal or proneural cells. And the proneural cell seems to be more active at the beginning, but then their activity drops down rapidly, and the more sustained effect is attributed to mesenchymal cells, the ones that have mutant EGF receptor. So in fact, the presence or absence of these different cargos, including the oncogene, may have quite a potent effect on how endothelial cells and vasculature responds to these stimuli. So I've been sort of focusing my comments on EGF receptor primarily because this is a very paradigmatic molecule and a potent oncogene, glioblastoma. But of course, we all know that there's more to it. And in fact, EGF receptor is a part of a signaling pathway that we all know all about. And this pathway includes a number of potent proto-oncogenes, one of which is RAS oncogene, which is mutated in a very large percentage of human cancer, about 20 to 30%, which obviously makes it an important oncogene in the grand scheme of human malignancies. So can we apply some of the same lessons or some of the same questions that we've just explored with EGF receptor to mutant RAS? And to be able to do this or explore this question, we've used a system whereby very non-transformed epithelial cells called IEC18 who have been engineered to express mutant RAS. And we simply ask questions about whether or not this will change their vesicle production, how this changed the vesicle activity, and so on. And the first thing we've noticed was rather startling increase in overall production of vesicles by these RAS transformed cells. So this is just an NTA plot showing that RAS transformation alone increases numerical output of vesicles by these RAS transformed cells vis-a-vis their non-transformed counterparts. And in fact, as in the case of VEGF receptor, there's a shift in protein content, with some proteins being almost exclusively present in vesicles from RAS transformed cells. And among those exquisite cargos, there's of course a RAS protein, mutant oncogenic RAS protein in itself. And these vesicles also contain RAS, mutant RAS mRNA, as well as DNA, interestingly enough, in fairly large quantities. So what do these, so RAS is a very potent oncogene, and you know, this is an oncogene that sort of almost led to the discovery of oncogenes per se, because it was so potent, and it was able to transfect or transform the cells, normal cells, to which it was introduced by, you know, different methods of transfection. And the question is, can we then take vesicles from RAS transformed cells and transfer RAS into normal epithelial cells, rendering them malignant or altered or transformed? And to our great surprise, the answer is no. And the simple reason for this is these cells, they are relatively inefficient in their vesicle uptake. And this is a feature of several, not all, but several different non-mesenchymal non-transformed cells, that they simply are not particularly good at uptaking vesicles from cancer cells. However, mesenchymal cells such as fibroblasts are great uptakers of RAS vesicles. In fact, they respond in a very dramatic way by increasing foci formation as if they were transformed by mutant RAS expression, but this effect is not permanent, so the integration of RAS doesn't occur, and the cells eventually lose their colony-forming efficiency. Similarly to EGF receptor, treatment of endothelial cells such as HUVECs with vesicles from RAS transformed cells stimulate growth and migration of these cells, which sort of manifests certain angiogenic activity that these vesicles may have. And so we were very intrigued by this. EGF receptor causes sort of angiogenic-like responses in vesicles and also RAS. And we're trying to understand what really is the nature of this response. How do endothelial cells perceive this interaction? And there was a number of different steps and missteps we have taken. But at one point, we began looking at the genetic makeup of both IEC18, these normal cells, and the RAS counterparts, using different methods. And we found that there's a dramatic difference in the genetic composition of these cells. So IEC18 cells are fairly euploid. They generally have only 2 chromosomes of each pair, and they have a fairly normal genome. On the other hand, introduction of RAS into these cells essentially explode the genome of these cells. They become very abnormal. They contain multiple copies of different chromosomes, in this case 2 and 3, and finally 1 and 2, and finally, they form very obvious micronuclei, which are very easily counted in cultures of these cells. So we thought, oh, you know, these cells are genetically unstable. Is this going to impact in any shape or form their interactions with different target cells? And the other thing that we've noticed, and this genetic instability of RAS transformed cells is associated with a very sort of interesting and enhanced deposition of intracytoplasmatic chromatin, which can be labeled with BRDU, and we found that this includes or leads to emission of some of this DNA into cell exterior in the form of extracellular vesicles, and this process of emission of extracellular vesicles containing this genomic DNA is in fact a function of autophagy, because when we block autophagy with chloroquine, the amount of DNA being produced was dramatically decreased, and this is now documented in the number of systems. But in our case, it'll allow us to understand that some of the processes of DNA emission, including the oncogenic DNA, are potentially could be autophagy dependent. And so what would then these highly abnormal vesicles that contain DNA, RNA, and RAS protein and many other different proteins, how would they impact endothelial cells? First of all, we've noticed that they are readily uptaken by endothelial cells. And when we started looking at these endothelial cells, we found that untreated endothelial cells, of course, had a very normal genome. They were primary endothelial cells, HUVECs from normal blood vessels. But when we treated these cells with vesicles containing all these different RAS-driven cargoes, we found that these cells, the normal endothelial cells, as it were, to our great surprise, started producing micronuclei, which suggested to us that they may be actually, in fact, genetically unstable. And this can be seen also through DAPI staining, where these little dots appear in endothelial cell cultures. And we quantified this, and it turns out that there's about 3 to 4 times greater presence of these micronuclei in endothelial cells treated with RAS vesicles as compared to IC18 vesicles, which hardly change anything in terms of the endothelial cell behavior. And we don't know why that is, but part of it is that RAS vesicles contain much greater amounts of genomic DNA, of course, including RAS DNA, but they also contain a very unique repertoire of proteins. And when we compare different cell types, which can produce micronuclei in endothelial cells, we find that 3 different completely unrelated cell lines have at least 19 common proteins listed here, which may suggest, and some of these proteins include centromere-related proteins and other DNA binding proteins, so which suggests that there may be a certain repertoire of deregulated proteins in donor cells, which is then passed along to recipient cells, causing genetic instability. And you may say, well, what does it have to do with anything? We know that endothelial cells in cancer are normal, but they aren't. So although this is not a widely studied subject, but Michael Klagsmoor's group, many years ago has pointed out that in many human tumors and in xenografts, there is evidence that certain populations of endothelial cells actually become aneuploid and they start resembling tumor cells in terms of their genetic makeup. So how this impacts angiogenesis still remains to be studied. For the last few minutes of my comments, I'd like to focus on, I'd like to expand this discussion beyond the local tumor conditions and talk about systemic effects of oncogenic EVs in the context of hosts carrying the malignant tumor. And of course, there's a considerable number of systemic events that we know are attributable to vesicles emanating from cancer cells. For example, thrombosis. There's 2 cancer patients experience thrombosis, and there's, of course, evidence for thrombosis-inducing vesicles, and the same goes for hypoxia, activation of stroma, immunoregulation, vascular permeability. Of course, pre-metastatic niches is a paradigm for this sort of interaction, and also anti-metastatic niches. So there's many events that EVs are known or thought to trigger systemically beyond the confines of the primary tumor. So to be able to do this, EVs would have to somehow interact with cells in a systemic scene or sphere, right? So what cells do they interact with? And the first cells that they probably encounter, we thought would be the blood cells, and of course, apart from endothelial cells. And so we wanted to know where vesicles, what these vesicles are in a sample of blood of a mouse that carries this RAS-containing tumor. And so we fractionated blood to different fractions and tested the amount of RAS DNA as a sort of a marker for where the vesicles might have gone. And one thing that we've noticed was that, as expected, perhaps slightly anticlimactically, we've noticed a certain amount of DNA in soluble plasma, as we name it ctDNA, as in human context, but also in vesicles. So of course, the amounts are relatively comparable, maybe slightly more in vesicles. But what we were completely unprepared to find, we thought maybe platelets would have some vesicles, and we didn't know anything about RBCs, but we were completely unprepared to find that the vast majority of this signal per sample of blood of oncogenic DNA we will find in white blood cells. And in fact, so much so that white blood cells in these mice contain more oncogenic DNA than any other cells in organs that are normally seen as traps of vesicles. So we all know that when we inject vesicles into mice intravenously, most of them will end up in the liver, but liver didn't have any anywhere close the amount of oncogenic RAS as measured by droplet PCR as did circulating white blood cells. So that was very puzzling. And so we wanted to know what kind of white blood cells they are. And of course, we've done a whole bunch of flow cytometry fractionations. But one thing that we also did is we decided to suppress the population of neutrophils because they are numerically dominant in the white blood cell population, and see what would happen with the distribution of the oncogenic DNA in blood. So this is the untreated, on the left hand side are untreated mice, and on the right hand side are mice treated with anti-LY6G antibody. And as you can see, of course, white blood cells have lost considerable amount of DNA because there's not a lot of them. But what this did is also increase the amount of both free and vesicle-associated soluble DNA, meaning that neutrophils. There's two interesting messages I think this experiment taught us. One is that neutrophils, in a way, control the amount of EV and soluble DNA in plasma, at least in this model system. But there's also a notion that these neutrophils can actually contain or accumulate this DNA. And if that is so, and of course we are studying this now, with great diligence, leukocytes could actually become another platform of liquid biopsy. So we know about circulating tumor cells and DNA and circulating vesicles and educated platelets, but we also find that for certain cargoes, such as DNA, leukocytes may actually have a considerable advantage and may have notable amounts of this material, and perhaps usable for diagnostic purposes. And it is important to think of this process as a biological regulation rather than passive uptake. So for example, when we take leukocytes and we add free DNA to them, nothing gets uptaken. But when it's sufficient, we take this DNA in the form of nucleosomes or EVs, and then the DNA becomes uptaken. So there's certain biology attached to this, and we now know from the beautiful work from Jackie Goetz is that there is a pathway whereby circulating tumor vesicles don't just randomly distribute themselves to different cell populations, but they actually interact preferentially with myeloid cells and endothelial cells. And I guess some of our observations are somewhat consistent with that study. So the final comment I would like to make around the question, so what? So let's say leukocytes take up vesicles. Yeah, maybe they'll be usable for liquid biopsy, but what's the impact of this material on biology? And so we don't know yet, of course, the full spectrum of this, but because we are interested in vascular events in tumor progression, we looked at two different processes. One is the expression of the procoagulant receptor known as tissue factor, and the other one was production of inflammatory factor or chemokine known as interleukin-8. And interesting enough, ingestion of RAS-dependent or RAS-derived vesicles upregulates both of these factors. So you can argue that potentially in some systems, oncogenic programming of vesicles emanating from tumor cells may produce a thromboinflammatory syndrome that is actually seen in cancer patients and may complicate the outcome of the disease by promoting inflammation, thrombosis, and other similar events. So this is something that I guess remains to be studied because, of course, this particular influence is not opposed. To summarize, I attempted to survey some of the known some more or less studied aspects of oncogenic influence on the vesiculation process and communication. And I mentioned that the oncogenes have an impact on the release of oncogene. They reprogram cargo vesicles. They influence biogenetic machinery. They influence the repertoire of EV subset. They control, to some extent, transferability of vesicles or their interaction with target cells. And finally, that they may control aspects of extracellular vesicle uptake. And some of the thoughts that I wanted to articulate at the very end to just summarize what I attempted to convey is that they are shown here. So in cancer, the multiple pathways of EV biogenesis are altered by genetic and epigenetic factors, which may both be oncogenic and maybe involved in oncogenic transformation, disease progression. Cancer EVs from even uniform cellular sources are highly heterogeneous. They transfer oncogenic cargo between cell populations, and they may cause transient types of transformation, like states in target cells. They may produce angiogenic activity, but this angiogenic activity is not normal. It's not the kind of activity that VEGF would produce or other angiogenic factor. It actually may involve activation of genetic instability or DNA repair machinery in endothelial cells, rendering these cells abnormal. This is not the normal response of endothelial cells to angiogenic stimulation. The oncogenes, of course, impact multiple facets of EV-mediated cellular communication, and there's an important aspect of vesiculation and EV biology that involves utilization of these processes, liquid biopsy, and finally that some of this liquid biopsy biology could be actually, it could involve sequestration of material from EVs by leukocytes, and this may actually produce, or we think that this could be usable as another form of liquid biopsy, which we call leukobiopsies. On that note, I would like to thank you for your attention and apologize for some of the technical glitches. And if there are any questions or comments or yawns or other things, then I'm happy to take them. You cannot throw objects at me thanks to Zoom. So
thank you, Janusz. This was wonderful, wonderful presentation as always. I do have a few questions myself, but let's try to go through everybody's question. And I think we can start with the Kalyani. Are you online? Yes. Kalyani, do you want to ask your question? You might be muted.
Okay. So thank you, Janusz. It was a wonderful presentation. So my question is that has the EV-mediated transfer of EGFR between EGFR positive mesenchymal glioblastoma stem cells and EGFR negative proneural stem cells documented that can favor the pro-tumorigenic behavior of the proneural cells?
Yeah, that's an interesting question. No, it has not been studied to any depth that I would know of. On the other hand, what was studied is the EV-mediated communication between proneural and mesenchymal cells. And there was a study from Sandra Breakfield's lab and her colleagues, and they noted that both cell populations are actually altered through this interaction, right? So I think Nino Chaka was involved in this, Franz Riklefs, and so on. So there was a very beautiful paper in Cancer Research some years ago where it was found that these proneural and mesenchymal cells, which may sometimes coexist, they actually do interact by vesicles. EGF receptor was not implicated in these interactions, but it may have been involved. You know, this is something that would need to. So we have a project on that question in the laboratory right now. You know, having access to isogenic knockouts of EGF receptor and so on. So this could be explored more deeply, but no, I think, I mean, unless somebody knows of a study that I missed, but I don't think in the stem cell literature there was this kind of an experiment reported.
Okay, the next question from Reginald.
Hi, thank you for the very interesting presentation. Some of the first data that you showed where the EVs were uptaken by the fibroblasts and it caused the focus formation type of phenotype, and you said that was only a transitory condition. Is that because I assume you only treat it, you know, once with the EVs? So I'm just curious if there's like a point of no return where the buildup of the EVs will induce a permanent state change in these fibroblasts and even in the endothelial cells based on this genomic instability that could occur.
Yeah, you know, so I think genomic instability is definitely a possibility. We this experiment was designed to answer the question whether or not transfer of mutant RAS will lead to expression of mutant RAS in recipient cells by way of genetic integration. So can you actually use EVs as a sort of natural biological lipofectins that may allowed cells to acquire mutant oncogenes, you know, sort of in a lateral horizontal fashion. And we never found that. We never, like, I'm not saying it's very hard to rule it out, but we've upped the ante of this experiment considerably using quasi-toxic concentrations of vesicles to, you know, multiple additions and very high levels. And we've never actually seen the integration. But what would happen eventually is that these normal cells, fibroblasts, would actually become toxically injured by high levels of vesicles, and they would basically lose their viability. And you know, it is possible. So, and so we've done a very long-term experiment with endothelial cells to answer the question you raised quite rightfully and correctly. And so we thought, well, you know, is it possible that primary endothelial cells such as HUVECs, which have no losses of tumor suppressors and this and that, so there should never be any background of transformation of these cells, right? Never. With fibroblasts, you can always argue that maybe these cells spontaneously transform vesicles or no vesicles, right? But with normal endothelial cells, this would never happen, right? And these cells were grown for months and nothing ever happened. So, you know, we did all kinds of, you know, maneuvers with them. There's a paper on this in Oncotarget, when Oncotarget was still a journal with impact factor and listed. So, and, you know, we described all our, you know, trials and tribulations on that note, you know, in that paper. But to my, so of course, you know, this is not to say that stimulation with vesicles is never going to transform the cells, but I think one has to be careful once you explore this notion, because stimulation of cells with inflammatory factors or with growth factors would also transform them. So vesicles may not be necessarily, as an unspecific, toxic factor, may not be necessarily very unique. What is unique about vesicles is the capability of transferring material. But so of course, you know, it is the fact that the cells become transformed, let's say, in the natural history of tumors, is also a function of neighboring cells. For example, we know that there are cancers that grow in inflammatory environment, let's say in the skin or in the intestine, and these inflammatory cells effectively cause these cancers, right? But and maybe vesicles are involved, but this is a different process than horizontal transformation, right? So anyways, I mean, the debate goes on. We have a colleague here in our institute who is a great believer of horizontal transformation by way of transfer of DNA. And so we had a debate, well, we had a private debate in my office at one point, and everybody survived, right? So it was not toxic, but, you know, but there are people who feel that this may happen in some instances, you know, and we have never seen it, right? That's the only thing I can say.
So next question, I cannot wait anymore. I'm going with one of my questions, and next is going to be Lucia Languino. So my question, Janusz, I'm actually surprised that with all of our interactions, this has never come to my mind. You showed the HRAS-induced vesiculation and the fact that there is increased shedding of EVs measured with NTA. I'm wondering if you ever used other tools to look also at larger vesicles to understand if these oncogenes can also really change the shed of large oncosomes as well.
No, you know, not to the extent you would expect me to, right? And I'm very embarrassed because, you know, knowing you for many years and being a great admirer of your work, we should have looked at this, but we haven't. And for oncosomes, for example, your large oncosomes, I think, you know, NTA is just not adequate to measure this. So I cannot say that they are not there. All I can say that we've never actually looked at this. And with some of your expertise, I think we'd be very happy to fill that gap, because, you know, it's an interesting and important question. You know, I think in the grand scheme of things, when we know more about vesicles, we have to start dissecting their different functions. So I think we should sort of move away. You know, I'm just preaching now, right? So, but let me preach for a moment. I think we probably should move away from this overall descriptive term vesicles this, vesicles that, but kind of look at the populations and try and understand what these populations specifically doing. And this speaks to this.
Because they're very diverse. We know that now. Yeah, that's interesting. Thank you. So Lucia, you're next. Are you still there? Are you unmuted?
Yeah, I'm here. You want me to talk? I didn't know if you were going to ask the question. Okay. Hi, Janusz. First of all, super great talk. And I guess all of us cancer biologists really learn a lot here. So we've been promoting the idea that those vesicles are signaling compartments, but I don't know if the adapters come with the receptor or they have to be used, they are found in the recipient cells. For example, all the various Shek and all those molecules, are they found in the vesicle? Should we assume that they're there? Okay, all of them. Okay.
So we have certain limited insights into this, you know, partly because we've used some of these phosphoproteome, you know, antibody arrays and all these sorts of things. They're not sort of super reliable, but you know, they certainly visualize signals such as, you know, for example, MAP kinases and that sort of thing. And the answer, yeah, the answer to this, the answer is yes. Vesicles, at least in theory, do contain downstream targets of these, you know, membrane oncogenes or RAS. So they contain, you know, phosphorylated MAP kinase, AKT, STAT, and so on, right? So it's possible that these, as you put the signal compartments, are actually transferred en masse, so to speak, to the recipient cells and you know, interact with some membranes and then activate the whole signaling cascade. Or it is also possible that they may engage some of the, you know, internal or recipient cell targets. So, you know, I think it'd be an interesting experiment to try and dissect it. So there are now these, you know, CLAC or SILAC proteomic approaches where people can actually distinguish between proteins coming from different cell sources and see which get phosphorylated and so on. And you know, I'm blessed with having a proteomics guru, Dong-Sik Choi, in my lab right now. So maybe I'll talk to him about this. But you know, I mean, yeah, absolutely. I think this is a terrific and important question.
Perfect. Thank you. And thank you, Dolores and Carolina, for organizing this. But great talk. Thank you.
Thank you.
So one other quick question I have is, so you showed the micronuclei and the cytosolic DNA in your systems, and I also find that incredibly fascinating on, you know, how this DNA is enclosed in vesicles. But one thing I'm wondering is, how do you really quantify the micronuclei by DAPI staining, and how can you distinguish this DNA from mitochondrial DNA? Because I just read a paper where they use a very similar approach, but they conclude that, and they only look at mitochondrial DNA, and they conclude, and this is a Nature paper, and they conclude that there is only mitochondrial DNA in EVs, which I know is not the case.
You know, well, you know, this is I know that both you and I have been embroiled in this debate over vesicles, and of course there's you know, Bob Coffey's and Danish Jeppesen paper, which sort of critiques the whole notion of exosomal DNA and that sort of thing. And you know, I think it could be actually linked to what kind of cells people use and whether or not some of these things are generalizable or not. So why is it not a mitochondrial DNA in vesicles that we study? It is simply because we've sequenced this, okay? Mm-hmm. And so we've sequenced this, and again, this is one of my, you know, low profile paper nobody reads, but you know, and that's why you know, I didn't get a lot of traction with these things, but we've actually done the whole, you know, whole genome sequencing, and there was exome sequencing, and something like 95% of the whole genome is actually present in vesicle preparation. So it cannot be mitochondrial DNA, because this would never happen. And likewise, I have never actually heard of RAS gene being present in mitochondrial DNA, right? So we have pretty good bioinformaticians, and they would probably, you know, pick up if there was a strong or enriched mitochondrial signal. Now, of course, there's a notion whether the DNA is on the outside of the vesicles or on the inside of the vesicles. I mean, you know, and the standards for proving this are changing, right? So when we were more active in that particular sphere, we would do all the digestions with DNases and so on. We would disrupt vesicles and digest them again, And when we disrupt vesicles, DNA was gone. When we didn't disrupt them, DNA was there. So, you know, it may well be that DNA is protected on the surface of vesicles. And I personally don't have a particular problem with this, right? I don't have an obsession with the luminal versus abluminal, because what actually interested me about this is the fact that this DNA actually can get transferred and interact with either DNA repair machinery in recipient cells or with the genome, or may change, you know, may mop up some of the effectors of epigenome and so on, and wreak havoc in recipient cells. So whether it's external or internal, you know, it may make a difference, but it's not essential to determine this. But yeah, no, I don't have any any you know, conclusive smashing evidence that would match the Nature paper to prove my point. But except for the fact that for the systems we've analyzed, you know, we're pretty confident that what we see are non-mitochondrial sequences, right? Of course, we don't have the money to sequence every single, you know, EV preparation we make, but you know, yeah.
One thing, it could totally be dependent on the system, as you have already alluded to, and maybe cells with chromosomal instability have this capability and other cells do not, because I think this paper was on benign cells, so it was not. I'll send you the paper anyway, because it's interesting.
Please do, that would be terrific. And you know, this is an excellent point, because when we look at cells that have relatively more quiet genome, right? Some of these mismatch repair mutant colorectal cancer cells, the amount of EV-associated DNA is considerably lower, right? So you have massive clastrogenic events in cells. This tends to send a lot of chromatin into the cytoplasm, and of course, you know, this then becomes somehow engulfed in vesiculation apparatus. But yeah.
We have a last question, if he's still here, from Tarun Tyagi. Tarun, are you still here? And unmuted. Yeah, Tarun, go ahead.
Hello.
Yeah.
Hi. Yes, I'm here. Yeah. Thank you.
Go ahead.
Hello.
Yeah. So I just want to ask you, these EVs, tumor EVs, when these are taken up largely by neutrophils, the circulating neutrophils, as Dr. Rak mentioned. So have you also checked their phenotype, if they are different in some way?
So we checked their phenotype with respect to very selected features. So we were interested, for example, in whether or not these neutrophils, which, you know, as you may know, white blood cell count is a risk factor for thrombosis, right? And gliomas are associated with high thrombosis. So we thought, well, maybe in some of these cases, not just neutrophils, but let's say white blood cells may have a role in this phenotype. And so we tested them for tissue factor. And tissue factor was, the activity of tissue factor was actually very dramatically upregulated on these cells that have taken up vesicles. And so was expression of interleukin 8. So we looked at a whole bunch of cytokines and chemokines using one of these, you know, I don't know however many analyte platform. There was like an antibody array. And interestingly enough, there was not all that much change except for interleukin 8. I don't know why interleukin 8 is affected to this extent. And so we have not looked at things, for example, related to antigens or proteome or that sort of thing, which we probably should have done, but we didn't. So this is an open question for anybody to explore. I know that, for example, people have looked at uptake of vesicles by myeloid cells, and they've looked at immunosuppressive capabilities, and those have changed. And so I think it's a very reasonable question, and one worth exploring. What is the global effect of the phenotype, you know, that may be imposed on these cells by the uptake of vesicles, right? I think this would
absolutely. Thank you.
We actually have a very last question. I promise this is the last one from Shivram Krish.
Yeah. Hi, Dr. Rak. Very nice talk. So I have a question. Do you think these vesicles have a inherent sense that which cells they want to transform? So like, is it that RAS3 within tumor microenvironment, it has some priority that it want to go to which cell type, like cancer cell type or endothelial cell or fibroblast cell or WBC? Does it has that sense? These vesicles have that sense, or it transforms everything that comes its way?
Yeah, so I think it's an interesting question. I don't think well, the vesicles are just basically just bits of cells, right? So it's kind of funny you say this, right? Because I was just about to say that they don't have any motility, so they cannot find anything. But I have some data that I cannot necessarily talk about where there is potential that may actually have motility, but I think the recognition or the interaction between vesicles and target cells are defined by the ability of these vesicles to interact with the surfaces of these cells and the ability of these cells to either respond to these signals or internalize these vesicles. And these are not random processes, for example. So we find that, for example, transformation with RAS oncogene fundamentally changes how the cells uptake vesicles, right? Fundamentally, it's a completely different mechanism for non-transformed cells and transformed cells, right? So, this is what the difference is, that, and white blood cells and endothelial cells, they have they're pre-programmed for this, for uptake. Endothelial cells transmit a lot of things through their own bodies, so to speak. And of course, white blood cells are phagocytic. So the fact that cancer vesicles spontaneously interact with these populations is probably, to some extent, a function of this receptiveness of these particular cells to vesicles. But of course, we know about integrins, how integrins sort of target vesicles to specific organ sites and so on. So there's definitely more to it. I think there's definitely a process or science of this targeting and interactions, but I guess this would be best answered in a specific case, right, rather than in a global sense.
Yeah. Thank you so much.
Okay, I think we're done. This was great.
Thank you very much, Dolores. That was wonderful moderating the session, and Janusz, wonderful talks. And I think we learned a lot about how EV mediated the communication between various different cells, and then how the endothelial cells also supporting the development for tumor using the EV. So I think that concludes our sessions today. And next week, we will have Lenara Balaj to give a talk on the plasma EVs. And on Thursday, we'll have sessions from the Europe with Irina Nazarenko to give a talk. So I hope I see you in both sessions, otherwise next week. For the moment, please stay safe. I know in US it's not very quiet right now, so I hope you all take care of yourself and keep away from all that crazy situation.
Yeah.
Thanks, Carolina. Thank you so much.
Thank you so
nice
much.
seeing you, everybody. Bye.
Bye, everybody.
Bye.
Thank you. Bye.